Welcome to Talk+Water, the podcast that takes a deep dive with those making waves. My name's Todd Votteler, and I'm your host. I'm also the editor-in-chief of the Texas+Water Journal and Texas+Water. I'm here today with Dr. Dean Hendrickson, the ichthyology curator at the Biodiversity Center at the University of Texas at Austin, for the University of Texas at Austin. And we are going to talk about, among other things, fish in aquifers and other life forms in aquifers. And so, Dean, welcome.
Thank you. Glad to be here.
So let me start off by saying you are finishing up your margarita, which was a margarita on the rocks, right?
Correct.
Okay. And we're here at Waterloo on Slaughter Lane, and I'm drinking a Yingling. And so I really like Yingling, and, discovered it, I don't know, maybe, 10 years ago or something. But at the time I was in Tennessee, and they told me, well, they don't sell it west of the Mississippi. And so, you couldn't get it up here for a few years after that. And I think, finally, now they're selling it. So, and I'm sure, everybody wants to know about that. So, Dean, you and I have known of each other and of each other's work for a long time, going back to the Edwards Aquifer work I was involved with starting in the 90s. Why don't you tell us a little bit about yourself, but start out, if you would, with how you became interested in water and all the amazing things that live in water.
Well, I guess basically I grew up in Arizona, in Phoenix, obviously in the desert. And every summer my parents would take me and my sister up to a little cabin they had on a trout stream in the mountains, about a mile high, a couple hours from Phoenix. And so I grew up spending every summer of my life up there on that trout stream, got into fishing, actually was busted by a game warden once for having taken more than my limit, but I did that because I had actually bought a license before I was required to, so that I could catch more fish. And he busted me saying, no, that's not possible. You can't have a license until you're 14 years old.
Oh, funny.
So I was deep into fishing, probably by the time I was about 8 or 9.
Are these rainbows or
these were stock rainbows? Yeah.
Okay.
Yeah. There were native trout in Arizona, but not the area where I was fishing. These were all introduced rainbow trout. But anyways, that got me into, basically that was my first introduction into critters in streams, basically. And so I eventually picked up fly fishing. That got me more into the interactions between all of the invertebrate fauna in the stream, stuff like that. And I had a very influential uncle in my life. He was a biology professor. At the time I was growing up, he was in Southeast Asia. But they would come through every year on tour, and they would stay with us. And he would go out fishing with me. And so he got me much more into the details of all of the aquatic fauna in the stream, stuff like this. So I think that was what really set the stage for becoming a biologist. And we did lots of experiments. He would show me what mayflies were coming off at the time, and we would try to tie flies that mimic them and that sort of thing. So lots of experience fishing and being outdoors. And then I guess that continued through high school, definitely, and actually my undergraduate years too. I was probably spending more time hunting and fishing and a little bit of doing alcohol and drugs and whatnot than I was studying, and accidentally, or not purposely, ended up on academic probation because of that. And then I had a very influential professor at Arizona State University where I was doing my bachelor's degree. I took his first the fisheries management class and then his ichthyology class. And he kind of took me under his wing, and it suddenly clicked with me that, my hobby could become my job. I could really get a job. Somebody would pay me to do what I love doing.
That's a recipe for a happy life.
Yeah, it worked out great. So that literally turned me around, and I graduated only one semester behind schedule, and then I went straight into the Peace Corps. Actually got married first, right out of bachelor's degree, and we went together to Colombia, and I was a fisheries biologist for the Colombian government. They were working on a United Nations funded project into the Rio Magdalena. Incredible fish diversity. It just really sucked me in, and that was it. That was, okay, I'm a biologist. I'm always going to be a biologist. And then eventually ended up here at University of Texas after a spell back in Arizona as the state endangered fish biologist. And after a spell in London where I did a master's degree in applied hydrobiology, which was interesting because it really, going from Arizona through the tropics to London, I'd seen the planet's spectrum of the way water is managed, basically. And then came back to Arizona, was doing my doctorate there, and got a job as the state fish biologist on the side. And so that got me into the bureaucracy of conservation management, and basically all of my research ever since has been related to conservation and endangered species. A lot of research in Mexico after I came here and a little bit before. So a long time focus in Mexico, but now pretty much focused in Texas.
Well, if anybody wondered whether you like fish or not, I mean, your shirt, you got a shirt on here with it looks like 100 different fish. And so that was a dead giveaway when I was, trying to figure out. That was
the clue. When he said, yeah.
Which person here is Dean?
That's funny. I never even thought about it. But yes, I have some fish T-shirts.
That's probably not shirts. The only shirt you have that's got fish on it, I'll bet. So tell me about the Biodiversity Center and the collection you have. You got species from all over the world, or scientists, taking them out and looking at them and trying to advance this state of knowledge about the various individual species. I mean, tell me what is going on there.
So I was hired here in 1990 as the curator of the ichthyology collection, which was then part of the Texas Memorial Museum, a natural history museum here on campus.
That's the one that is, I think I've been to.
Yeah.
They've got, dinosaur, bones as well. My daughters love that museum, by the way.
It's a great little museum. Unfortunately, it's not very well supported by UT right now, and its future is a little bit up for grabs, but lots and lots of people in Austin know it as exactly that, the Dinosaur Museum.
Right.
Tons of school kids going through there on ditchers. It's a great place. It really is. Yeah. But what most people don't know about that museum is that it was the one that had these scientific collections, which were all behind the scenes. The public didn't get to see them. UT has large paleontological collections. Some of those provided dinosaur skeletons that were on exhibit. And
we always have to have a leaf blower. Yeah, I was like, when are they going to have? I saw an advertisement for somebody who's got a rechargeable one.
Yeah, right.
Yeah, we're good. I think right now. Yeah. Thank you.
So, anyways, the Texas Memorial Museum became the owner of all of these scientific collections. Lots of specimens of plants, fish, insects, you name it. A large chunk of biodiversity. Preserved specimens or dried specimens in the case of the herbarium, the plant collection. But all behind the scenes. It was very not public research collections that scientists were using and still do use extensively, both using the specimens as well as the data associated with the specimens. So what was collected when and where. They can reveal lots about the conditions of the habitats from which they were collected. So they're used to study changes in ecosystem over time. Very valuable resources, both for the data behind the specimens and the specimens themselves. They accumulate toxins. There's just all kinds of research you can do with these specimens. So that was my job, was to curate and help grow this collection of preserved specimens that my predecessor at UT, Dr. Clark Hubbs, had started.
I remember him.
Yeah, I'm sure you do.
So people will have the background of that story. Clark Hubbs was the professor who was, I think, essentially the star witness for the plaintiffs in Sierra Club versus Babbitt, the litigation over the endangered species in Comal and San Marcos Springs from the Edwards. And I remember Dr. Hubs, met him several times. And so this is his collection.
Yes.
And are you still adding to it? Are you still getting?
Yeah. In fact, I think when I came, it had something like about 10,000, 12,000 specimens, and we're now up to over 80,000, I believe, something like that. So it's been growing quickly. And a lot bigger now than when he left. Yeah.
So do you have researchers from, other parts of the country or other places in the world come to see these specimens and do some work with them and go back and write a paper?
We function just like a library. Basically, we consider our specimens to be books that are written in languages we're still learning how to read, like eDNA, or DNA is one of them, basically, that sort of thing. But that's what these people use them like, and we send out loans or they come and visit.
Oh, I was going to ask you, so it's like interlibrary loan. You actually send them out to another institution and then they go look at it there and it comes back to you, huh?
Yeah, we function very much like a library.
Okay. Yeah. And all these are usually, I'm going back to high school now, right? In a jar with formaldehyde or something.
Right, yeah.
Okay.
They're preserved in formaldehyde, and then we store them long-term in ethyl alcohol.
Okay.
Right.
So you and I think, our first kind of coming aware of each other was associated with the Edwards.
Yeah.
And what I remember was in 94, when I was starting to do some of the background work for Judge Mutton on this habitat conservation plan, that the interests involved in that litigation were going to draft. I came across, mention of these catfish, called blind cats, blind catfish, that were deep in the Edwards Aquifer. And there was almost no other information about them. And which I was like, well, are we supposed to be, should we be thinking about these other species when we're trying to come up with the management plan for the aquifer? So that's all I knew. And how much do we know about them now? Do we know much more?
There's still not a whole lot more known about them. These are probably the most difficult fish to collect on the planet, basically. They live 1,000 feet below the city of San Antonio. Their habitats are only accessible via wells. Humans can't get down there, so these specimens just occasionally. The first ones, I think the first ones came up in the early 1900s and were described in 1930, coming up through artesian wells. At that time, San Antonio was drilling these wells into an artesian water for aquifer, so you get a gusher.
Right.
And these would come up in those gushers. So those were the first specimens. That's how they were discovered. But then it sort of transitioned to this world in which there were many, many, many more wells drilled, but they all had pumps. And so they were probably getting chopped up by the pumps. The more recent specimens are all chopped up. So incredibly difficult specimens to obtain. And therefore, we just don't know much about them. There's hardly any specimens that are I think the number of Satan specimens, the wide-mouthed blind cat, is in the 20s. And the Trogloglannus, the more common one, is maybe in 50, 60, something like that. I can't remember the numbers off the top of my head. But that's the extent of the world's specimens of these things. There are probably thousands of them living under San Antonio. We don't really know.
And so there's the one that you called Satan. That one is predatory and it feeds off the other one.
Yes.
And the other one's the grazer, and it is grazing on the chemotrophic life, I guess, that's on the Edwards limestone and dissolving limestone.
Correct.
It's kind of like, it's not really, I guess, the same thing, but I kind of think about these smokers. What at the at the bottom of these trenches, and you've got all this strange life that never sees any light from the sun,
right?
And it's a really unique ecosystem. But these fish, so they've evolved where they don't have any eyes anymore, right? And so does that give us some clue about how long ago the catfish that they're related to entered into the aquifer and eventually became what it is today?
That's one thing that we can speak with some scientific knowledge about is at least one of these, the suckermouth catfish, Trogoglanus, we have gotten recent specimens of that were not preserved in formalin. Thus, we could get DNA out of them. So we sequenced that. We sequenced its entire genome. And that has just only in the last couple of years made it possible to include them in phylogenetic or evolutionary history analyses of the entire North American fish fauna. And so we can pretty accurately say now that these things probably invaded caves in Texas as the aquifer was forming, probably at least, say, 30 million years ago. Wow. Right about then. And so they diverged from their surface ancestors. Catfish, of course, all catfishes are kind of prone to go into dark places, right? So they were going into these caverns that were starting to develop, and these things found their way deep into the aquifer and over millennia lost their eyes and became highly adapted to that subterranean environment that lacks light completely. And there's a very diverse ecosystem down there that they can feed on now. Lots of invertebrates have evolved into the same system. And so, so they've been in that aquifer for many millions of years for sure.
And so they're just a couple, 2 or 3 inches long. Most
Yeah. Trogoglanus gets maybe to about 4 inches, and Satan gets to maybe 6 or 7 inches.
And so, I'm kind of thinking about this and wondering, is their presence a clue, or does it indicate that maybe there are channels that link these aquifers in that region, and maybe that they are connected in some way with populations maybe in Mexico? I know the Edwards kind of peters out over there, maybe.
Right. Yeah, there is a sort of an extension of the Edwards that does extend into Mexico, and that's where I first got into the blind cats. Basically, the Mexican blind cat lives in a shallower aquifer, but it is part of the Edwards aquifer. It's definitely connected to the Edwards aquifer, and they actually cross the border in that aquifer in the area around Del Rio. Most of my work has been in Mexico on them, and we always expected to turn them up in Mexico, and we finally did about 5 years ago.
Okay.
Their closest relative, apparently, is one of those that occurs under San Antonio, Trogoglanus, and they diverged from each other again like 30 million years ago or 20 million years ago. So there definitely is a connection between the aquifers and these fish, though, in Mexico. The Mexican blind cat is in a much shallower aquifer that we can access through caves. It's usually a vertical drop of maybe 60, 80, 100 meters down to what are basically just surface streams running through a cave.
Oh, wow. Have you done any of that? Have gone down there and gone diving and all that?
Yeah.
Oh, you lucky duck.
Yeah. It was a really cool project, a lot of cool explorations. So
people don't know. Scientists have cool eyes. I mean, depending on what you're doing, what you just described, that's it's pretty damn cool.
It was great fun. Yeah, it was really, really exciting. So, and then actually we kept those in the lab too, so we know a whole lot more about that species than we do about the two that live under San Antonio now, because we could collect them and actually study them in fairly rigorous ways. But I was going to say, kind of lost my train of thought here. So they're related to the Edwards aquifer ones under San Antonio, and we can make some inferences then, knowing a little bit more about them, about what the San Antonio ones are like. But getting back to these crevices, that's the really cool thing about the Edwards aquifer under San Antonio is it's coming, the water that in that aquifer comes out of the hill country, nice fresh water, and it flows down, and then at one point it runs into another aquifer, a very relatively saline, sulfurous aquifer, and where those two abut, basically, it's called the bad water line, typically. And you get this interesting chemistry that goes on that actively dissolves the limestone via bacteria that are feeding on it. Basically, the chemistry is producing energy that they can consume, and that's the basis of the entire food chain for the entire ecosystem now. So that's what Satan and Trogoglanus evolved into. Trogoglanus feeds directly on that bacterial fungal mat that is living off the rocks, and then Satan and is almost certainly feeding on Trochoclanus and all these other diverse invertebrates that are down there. So a very energy-rich aquifer, which is uncommon in cave systems. So you have this rich energy source, and you get this high diversity that has developed there, evolved there over millions of years.
So you mentioned the bad water line. During the litigation, there was a lot of discussion about that because that line is near San Marcos and Comal Springs. Mm-hmm. And there was some evidence that the line migrated during the drought of record, and that when flows started again in 56, that a lot of bad stuff came out of the springs initially, and maybe killed a lot of things. Yeah. And I never really could make head nor tail whether overpumping the aquifer really would cause that line to migrate or not. I know that the proponents and opponents of that theory usually cited the same study, which is not a good sign about it being clear. But do we know anything else about that bad water line? I mean, essentially, it's where the saline portion of the aquifer and the non-saline portion meeting, right?
Yes, correct. Yeah, actually, if the map of Texas and its highways in San Antonio, it's just barely, the bad water line runs just to the east side of I-35, pretty much. So that'll give you some perspective. But so I'm not a hydrologist, so I probably shouldn't be speaking about this, but I talked to a lot of hydrologists about it. And I think the consensus is now that concern over the bad water line was valid, and it certainly is possible to move it. So if you really, really pumped on the good side of the bad water line, the line would move. It would move inward.
And you draw it into the springs, and then that stuff would come out the springs and go down the San Marcos and the Comal.
It'd probably first show up in wells. And in fact, there are a number of monitoring wells right along the bad water line that were used to study that phenomenon. But the way the aquifer is managed right now over the last number of decades, it's been relatively stable, and it's been controlled so that it wasn't allowed to overpump.
Right.
And so, at least in the recent decades, the bad water line has remained relatively stable.
Yeah, we've got a very good plan in place now. And so I think the odds of something like that happen are pretty low now. Yeah, thankfully. But so, okay, you mentioned eDNA, which means environmental oh wait, hold on. I'm gonna see if I can remember. Ribonucleic acid. I can't remember.
Deoxyribose.
Yes, that's it. Okay, dang it. It's been a long time. And so tell me about that. You don't have to actually catch the specimens anymore. You can find their DNA in water.
Yeah, this I'm hoping turns out to be a new, easy, and very powerful way to actually study these organisms, because the big impediment right now is sampling for specimens. We know them only from specimens, and all the historic sampling was putting nets over well outlets to catch specimens. Anymore, you simply can't do that for any number of reasons. If you were able to do that, somebody's going to have to pay the water bill, and you'd have to get a permit to discharge the water to the surface. It's very complicated. Right. So it's basically been taken out of our toolkit. And so all animals shed DNA. Humans shed DNA. Our skin sloughs off. Every animal loses DNA externally, and that DNA then floats around in water. And if you take a very fine pore filter, you can strain out that DNA, extract it, and analyze it just like you would DNA from a tissue sample for a cancer or something, whatever. So you have to technically know what DNA you're looking for is one problem, but there are some ways around that. And I mention it because we don't know anything about the DNA of Satan. All Satan specimens were preserved in formalin, which makes it near impossible to get eDNA from those, or DNA from those specimens. So we don't have a target for that. But luckily, we were able to get some recent specimens of Trochoclanus, and before they were preserved, we extracted DNA. And so we've got their complete genome sequence. So we know exactly what sequences to look for to be able to identify DNA of Trochoglanus. So now we can take very small water samples from any well and extract DNA and analyze it. And conceivably, it's possible to get a list of all of the organisms that occur in that aquifer if you know what sequences are associated with what organisms. So we're just now starting to do that, and it looks like it's going to work. It's kind of a novel environment to apply this new tool. But we're hopeful that now we can continue sampling, not so much for specimens, but just for DNA, and be able to at least say, yes, Trochoglanus, at least, we're picking up its DNA. It must still be alive down at the bottom of this well.
Huh. So you're collecting that DNA, and you probably find DNA of things that no one is quite sure what it is yet. I mean,
exactly.
Right? I mean,
Yeah.
it can also be, I guess, an indicator to you that, maybe the ecosystem down there is really diverse and there's all sorts of things swimming around that we haven't documented yet.
Yeah, the Edwards Aquifer has been pretty well known for a long time to be, in fact, at one time it was proposed to be the world's most diverse subterranean aquatic ecosystem. There are, I think, over 100 invertebrates known to inhabit that dark ecosystem down there, the 2 fishes, and incredibly diverse bacterial community that nobody knows much of anything about. And we have been able to, with our very first eDNA analysis, come up with long list of bacteria. They're not associated with species, but they're associated with the major different groups of bacteria. So we get kind of a feel for the community structure of the bacteria. Same with the invertebrates. There's not a lot of sequencing that's been done on those invertebrates, so we can't identify individual species, but we can usually place them from known sequences of others in major groups and stuff like that. And with the fishes, now we've got DNA of Trochoglanus. We should be able to identify specifically it. Satan, we're hoping some eDNA sequence that is from the family that we're pretty certain on the basis of morphology Satan belongs to comes up, and then we can say, well, we know the sequence for every other ichthyolurid in North America. This is an unknown. It almost certainly is Satan. So, but it's truthfully kind of a shot in the dark, but we're hoping we come up with this unknown sequence and we'll be able to say it's almost got to be Satan.
Gosh.
Yeah.
So using eDNA, obviously you're identifying what's down there, but can that data give you any, I guess, clue as to the population, the size of the population, anything like that?
It can't really, at least not yet. In theory, it should be able to do that. And there is some literature from surface ecosystems that attempts to do that with varying levels of success. But the current state of the technology is that you can't really say it can do that. I'm sure in the future they'll nail that down. But right now, basically, it's just telling us that we can basically detect presence. Like if we get the sequence of the known sequence of Trochoglanus in our DNA samples, we can say that species is down there.
Okay.
Yeah, that's
gotcha.
About it. Yeah.
And so when you say down there, if they're 1,000 feet deep, is that like the same pressure that you would have in the ocean, even though it's in the aquifer and the aquifer's got caves and all that?
Yeah. Yes, it is.
Okay.
Yeah.
So they're not going to do too well when you bring them to the surface, or will they?
They do better than most fishes would do because over their evolutionary history, they've lost some of the anatomical structures that are most affected by those pressure changes. They didn't need them in that environment. So like swim bladders that most fish have that are what kind of blow up as you pull a deep sea fish up and it protrudes out its mouth. If you've been fishing and done that, that's not a problem for them. So actually, one of each specimen of the San Antonio blind cats did survive for some period of time on the surface, but those are only those that come up through these artesian wells. If they come up through a pump, they get chopped up and we just get fragments, basically.
Gotcha.
But no, the pressure change is not a big thing for them. Really. No.
So when we talked a month or two ago, you told me about a project that when I heard about it, I thought, I got to somehow be involved with that. A hydrophone that would go down in the Edwards Aquifer and listen for these guys, kind of like the same type of hydrophone that listens to whales, right?
Yes, exactly. Yep.
And so obviously everybody knows about whale songs and all that. I mean, what would you think we'd be hearing from these blind cats and other things in the aquifer? Just little clicks or something, or would it be more than that?
Right. So we don't know that the two blind cats produce sounds, but we do know that almost every one of their relatives, the family Ichthyologist, which is about 60-some species, they've been fairly well studied and their sounds documented. And what they do is they stridulate, it's called, their pectoral spines. Most people have caught a catfish, you know that they often have very strong spines that can wound you quite quickly. Easily.
I've been stuck a lot of times when I was a kid. Yeah.
And actually a lot of fishermen are familiar with this. Channel catfish, for example, probably the biggest game fish in the family. A lot of times when you catch them, their pectoral fins will be going up and down, and you can hear this kind of a croaking-like sound.
Right.
That's produced by ridges on the articulation at the base of their pectoral spines. And they make those sounds. They're used in mating and during breeding season, basically. So since all of their relatives do that, we're suspecting that these blind cats might do that too. And at least one of them, and I don't remember which, appears to have the anatomical structures to do that. And they sometimes use their swim bladder to amplify it. But in this case, these guys have both lost their swim bladders. They're not going to be able to do that. So again, we're somewhat shooting in the dark here. We're hypothesizing that they will produce sounds because all of their relatives do. And we can drop a hydrophone down a well bore. There are lots of them plenty big enough to do that. Let it sit there and see if we catch some sounds that sound like some of these other members of the family, which are pretty well known. So.
Interesting. Well, so what do we think the future is for these guys? I'm just kind of, thinking that, there are a lot of people who are heading off to places all over the globe to try to collect samples of fish and insects and all sorts of living creatures that no one has documented before. But, here we are in central Texas and, just 1,000 feet below the surface of one of the largest cities in the country, here's this whole kind of undiscovered ecosystem.
Yeah.
It's pretty exciting, but it's just kind of overlooked, isn't it? I mean, in some ways,
it is very much overlooked. Yeah, it's we know that it is remarkably diverse. There are quite a large number of invertebrate species that are endemic to the aquifer. They occur only in this aquifer, and there are quite a lot of them that still don't have scientific names. They might be known from one specimen or something. They're known to exist, but nobody's actually gone to the trouble to actually describe them yet. And there are new ones being discovered all the time still. So there are lots more new species to be discovered in this aquifer for sure.
So what's next for you? What are you going to be working on here going forward?
Right now we've got this eDNA project. We've done a little bit of very preliminary sampling. It looks like the method is going to work more or less. And so now we're busy trying to get access to wells where we will be allowed to sample. It'll just take a little bit of water, so it's not that difficult. And so we're looking for wells where we can sample, run some more DNA tests, and see what we turn up. Basically, we're still in the very early stages of doing this. The biggest impediment in the past has always been that we needed to get specimens. We've gotten around most of those obstacles now with the eDNA. We hardly need any water whatsoever. Just literally a gallon is probably going to be enough for us. So very small amounts of water, but it's still not quite as easy as I'm making it sound. These wells are obviously things that they're owned by a large number of individuals and water management companies, things like that. And lots of people are afraid of new endangered species being described and perhaps causing them more water management issues, this sort of thing. So I don't know. We're just working hard to try to get access to whatever wells we can, and we'll see what comes out of it.
We've been talking about San Antonio most of this discussion, but we're here in Austin, and we're over the Edwards portion of the Edwards.
Yeah.
What about this portion of the Edwards? Any thoughts about whether there's something similar below us?
This part of the aquifer has been better studied than the deep aquifer under San Antonio. It's more accessible because there are more springs up in this area. You can sample the fauna via the spring outlets. So there's quite an active community of people sampling that, and lots of interest in things like the salamanders, the blind salamanders that occur here. Mm-hmm. There are blind salamanders that there aren't any known from San Antonio, but they get close. They're in New Braunfels. They get that far down. And we're including them in our eDNA studies as well. Independent of that, there's another kind of eDNA study ongoing out of UT by a colleague of mine looking at these salamanders that occur in the San Marcos, Austin area. And they're getting their techniques down too. They've had some struggles getting that going, but it also looks very promising. We're taking a slightly different approach. I'm not going to get into the details of the methodologies, but they're designing primers to look for individual species. We're kind of trying to cast a broader net and look at all organisms, basically. They're targeting the specific ones that they've been studying, so they can say this species occurs here, but they're not going to be able to detect anything else with their methods. They're just focused on the salamanders.
Gotcha.
Right. Yeah.
Gotcha. Any thing that we might learn about the health of our aquifers by looking at these species that are way down below the surface of our cities? I mean, are they bioconcentrating things that we would be concerned about? Do you think? Is that possible?
Yeah, for sure. In fact, I think that's one of the things that might have been affecting Satan. It's the top predator in that ecosystem. And most people know the stories of things like eagles and whatnot that were affected by DDT. The top predators accumulate a lot of those sorts of things. And so Satan is probably likely to have been affected by that sort of thing. There's all sorts of stuff that we know is in the Edwards Aquifer, is in every water body practically, but they could have been accumulating over the years. And so they're basically, I often use the canary in a coal mine analogy. Most people have heard that saying. And basically, we can use these things as indicators of potential problems for us.
Right.
And there's certainly that potential here, especially with such diverse biodiversity. There's every one of them will tell us some different part of the story about what contaminants or other issues there might be in the aquifer, rather than having to test for specific contaminants and do water quality analysis, that sort of thing. The changes in this community over time would tell that whole story much better and with less effort. And but the difficulty, of course, is those specimens have always been difficult to obtain. But now maybe with the eDNA, that can also help us track the changes in the community over time.
So I've just about finished my Yingling, which is kind of like, that's an indication that either I need to get another one or we're going to wrap up. And so it's a weeknight, so that means we're going to wrap up. And so Dean, why don't you tell us how someone can find out about your research and also about the Biodiversity Center? Where can they go?
Okay, so the Biodiversity Center is actually quite new at UT, I want to say 5-6 years ago or something. It includes all of the biodiversity collections like my own, the herbarium, the insect collection, the herpetology collection, all of that, as well as all of our field labs are part of the Biodiversity Center as well. A large number of faculty are affiliated with the Biodiversity Center. The director is Dr. David Hillis, a well-known authority on the blind salamanders, for example. So we've got a very diverse faculty and staff working on this now. And it seems to be catching on, getting some funding, and we're looking forward to the future. It's certainly helping generate more research and get the word out about that.
So if they put University of Texas Biodiversity Center in their search bar, and
I think that should turn up the homepage, yeah.
Okay.
In my case, Hendrickson Lab is what I always tell everybody. Just look up Hendrickson Lab. There's a veterinarian that's also a Hendrickson, and that might turn up.
He likes labs?
Elephants, actually.
Treats labs?
Yeah.
No.
Yeah.
Okay. Well, that's great. Well, hey, thank you for meeting me.
Yeah, thank you.
And discussing your research. So let's go ahead and wrap it up there. This has been Talk+Water, and my guest was Dr. Dean Hendrickson, the Curator of Ichthyology for the Biodiversity Center for the University of Texas at Austin. So I want to thank our listeners for tuning in, and to say that if you enjoyed this episode of Talk+Water, please let us know by giving it a like. My name's Todd Votteler. Let's talk water again soon.